people will join us as we keep going. So welcome, good afternoon, good morning, wherever you are in the globe. It's almost good evening for me, but welcome to the Research Qualities and Outcomes webinar. The series this year is entitled Demystifying Research. And as I said, this is a webinar that's being put on by the Research Qualities and Outcomes Committee, the Rqo Committee at NSGC. And I am lucky enough this year to be the chair of the Rqo Committee, so I'm introducing this webinar series. So just a little bit so everybody can kind of orient themselves. So the Rqo Committee is a volunteer committee that is led by the NSGC board, and the charges that are set forth by the NSGC board for our committee are really centered around demonstrating the value of genetic counseling and generating the data for evidence-based practice. And so as part of our role within the Rqo Committee, we work to curate and develop resources for the NSGC community to assess the practice of genetic counseling and to conduct research that informs this practice. And so this is kind of the idea of this webinar series is really to address this kind of guidance and resources for the NSGC community. And so last year, some of you may have joined us, but we had the opportunity, we did something called Research Curious, and those webinars are available to view. They're recorded on the Rqo website. But that last year's was really centered around kind of arming interested genetic counselors and sparking interest in genetic counselors in genetic counseling research. And we went through over the course of a year kind of all aspects of a research project, from developing a research idea to implementation, design, and analysis. And so I would encourage anybody who didn't get the opportunity to view those webinars to take a look at those recordings. This year, we're really focused on kind of continuing to really engage our community in research, especially those individuals who may not themselves recognize that they are doing research at this time. And this is because, even though there are the roles that genetic counselors serve as really just continues to expand and diversify, many of us are still in some type of clinical role. And the dynamic nature of genetic counseling and the fact that the science behind it is always changing and evolving really lends itself to the fact that we are often doing research on the job. And so we really wanted to start in this webinar series to point out the research that we are already doing. So I'm going to hand it over now to Melanie, one of the Rqo committee members, to introduce our speakers. I have a few slides just to kind of speak towards demystifying research. So I'll click through those. I always do this, forget that I have slides. But we're pulling back the curtain on genetic counseling research. You can see we are not AV experts, and so our slides only get this fancy. But some of the things this year that we want to kind of explore are really demonstrating how research is part of our clinical days and jobs, and really even those of us who don't identify as research genetic counselors are often conducting research. Illustrate the diversity of genetic counseling research, and also a big aspect and piece that we heard last year during our webinar series is really understanding these avenues to create time for research, and then also continuing to direct genetic counselors to research resources. So, okay, I will stop sharing and hand it over to Melanie.
All right, hello everyone. Thank you so much for being here. And I have the privilege of introducing our panel of speakers today. First is Michelle Morrow. So she received a PhD in neuroscience from Northwestern University and an MS in genetic counseling from the University of Pittsburgh. She's worked as a clinical genetic counselor in pediatric neurology and is currently the coordinator of GeneDx's Gene Matcher program, which she'll speak about today. We also have Rebecca Littlejohn, who graduated with a Master of Science in Genetic Counseling from the University of North Carolina at Greensboro. She is assistant professor at the Department of Pediatric and Human and Molecular Genetics at Baylor College of Medicine, located at the Children's Hospital of San Antonio. She has worked in clinical genetics with mostly pediatric patients for 9 years. And finally, we have Elisa Blesson. So Elisa is a certified genetic counselor at the Kennedy Krieger Institute in Baltimore, Maryland. Elisa is passionate about providing genetic counseling to patients and families with neurogenetic conditions and neurodevelopmental disorders. She's actively involved in the training and clinical supervision of students in this field. She received her master's degree in genetic counseling from the University of Maryland School of Medicine. She's a member of NSGC, the Minority Genetics Professional Network, and the Maryland and DC Society of Genetic Counselors. So thank you all to our speakers, and I'm going to turn it over to Michelle to share her slides.
All right, let's see if I can figure out how to do this. All right. And are we looking at my screen now?
Yep, we're good. It's not in presenter mode, so you could switch it over
Oops.
to, there we go. And
Are
then
you
if flip
guys
it,
looking at presenter mode, or are you looking at?
we see presenter mode rather than the main slide.
Right.
I think there's a choice to flip display set.
Yeah.
There we go.
Sound good? All right.
Perfect.
Thanks for bearing with me. My name is Michelle Morrow, and I am a genetic counselor at GeneDx, where I've been for about 3.5 years, and I work exclusively in the Gene Matcher program as part of the research team. I always get really excited to talk about Gene Matcher because I think it's a wonderful resource for busy clinicians and busy genetic counselors to be able to dip their toes into research. And it's also a great resource for anyone who's working on the clinical side to kind of get more information about about what's going on with their patients and finding other resources to learn more about genetic variants. So Gene Matcher is an online platform that was created in 2013 as part of like a collaborative effort between Johns Hopkins and Baylor. And basically the way it works is that you can go to the Gene Matcher website, create an account. Gene Matcher is open to clinicians, to researchers. It's also open to patients and families, although there seems to be more traffic from the clinicians and researchers. You create an account and specify what gene you're interested in. Then you can also specify if you're interested in a particular variant or if you're interested in a particular phenotype. And then you just send it off into the ether. Once a matching entry is created by another person, then you will get an automated alert saying you have matched with so-and-so. Here's their email address, at which point you can go on and contact them and share more information. So this is a great program in general. And just to give you some idea of the scope of Gene Matcher, you can go to their website, and this is just a screen grab from the Gene Matcher website. Over 12,000 submitters, over 60,000 submissions, 15,000 genes practically, and practically 10,000 matches. This is great. And then as an aside, you'll notice that over 100 countries are represented. And one of the magical things about Gene Matcher is that it is a great way to get people from different parts of the world involved and hopefully expand the diversity of the population who gets involved in genetic research and genetic testing. So one group of genes that is particularly important in Gene Matcher, it's very well suited to Gene Matcher explorations, and it's kind of the reason that the GeneDx Gene Matcher program exists are called candidate genes. Let me see if I can forward. For some reason, I'm not able to forward. That's unfortunate. Let me see if I click. Nope.
Sometimes if you just kind of move, there we go.
Here we go. All right, candidate genes. Candidate genes are also called genes of uncertain significance, and these are genes that are hypothesized to be related to a disease or a particular phenotype, but we don't yet have enough evidence to say for sure that that association is real. We need more evidence. We need more scientific research, and that's where you guys come in. So of note, all variants that are identified in candidate genes are going to be reported out as variants of uncertain significance. If we're not certain about the gene's function, then we're not certain about the effect of the variant. And as you know, from a clinical perspective, variant of uncertain significance is, can be anything from disappointing to very distressing for families. And we really want to get those candidate genes, find more information about them, and upgrade or elevate them to a disease-associated status so that we can get more diagnoses for our patients. That's kind of what GeneDx's goal is in participating in Gene Matcher. What does it take to elevate a gene or to upgrade a gene from candidate to disease-causing? Research. We need case studies involving a group of individuals with consistent phenotypes, maybe a bunch of patients with a similar genotype. And then functional studies are also very important in kind of solidifying our information and increasing the confidence that there is a disease-gene association. So GeneDx is, as I mentioned, involved in Gene Matcher. We actually submit all of our candidate gene findings into Gene Matcher. And to give you an idea of the scope of that, I've got the little pie chart on the side showing outcomes from our laboratory. And you can see that almost 7% or about 7% of exomes come back with a candidate gene finding as the sole finding. And that's really a group of reports and a group of individuals that we have some agency over, that we can hopefully find out more information and change those variants of uncertain significance into a genetic answer for the family. And now I'm going to run into my there we go. Very good. So this is our laboratory's workflow, and I'm sharing this with you so that you know what the process looks like on our end and how you can get involved as clinicians. So as I mentioned, GeneDx submits candidate findings to Gene Matcher, then a match notification is created. And I sit in front of a computer all day and try as hard as I can to answer all of the inquiries that I get that come to us when people receive those matches. So they might receive a match and contact the GeneDx email and say, can you tell us more about your match? We have a small but wonderful group of genetic counselors. It used to be very small, but now we have a couple more people on our team, which I'm really happy about. We can assess those inquiries and find out what someone wants to know. Maybe they're interested in finding out what our phenotype is in our case cohort. Maybe they're interested in finding out if we have any de novo cases, if we've seen a particular variant. So we can look in our data, find out the answer. And if this individual who's asking is interested in putting together a research cohort, we can then tell them, sure, we can see if some of the clinicians who have ordered genetic testing from us would be interested in participating. And that's where I would reach out to you in the clinic with an email saying, we have a potential collaboration opportunity for you. Would you be interested? And we can offer the collaboration. Now, now the work falls into the clinical team's hands. The genetic counselor may reach out to the family, find out if they're interested in participation, because of course it doesn't go any further if they're not. If the clinical team wants to talk a little bit more to the researcher and find out a little bit more about what is going on, then we can facilitate that introduction, and we can then connect you to the researcher. Some time may pass, so you can exchange clinical and genetic data and perhaps fill out a spreadsheet, perhaps provide some other information that the researcher wants. Eventually, we hope that this ends up as a publication, but there's a little bit of kind of a black box between the time that you are introduced to the researcher and the time we hear about a publication. It can be 2 months, it can be 2 years. Sometimes it's as a case cohort is gathered. Sometimes it is because functional studies are being done. Many things can happen. So eventually we've got a publication. You as a contributing clinician will be offered an authorship space. And if on my end, if I notice that our ordering providers and genetic counselors are not being offered any authorship, we'll follow up on that and try to advocate for that authorship. But even after the publication is out, yay, publication, you get your name on a paper, hooray. Even after that's out, the work isn't done because we, on the lab side, will look at that publication and we will evaluate it to determine if this publication gives us enough information to upgrade the gene or validate the gene from a candidate to a disease-causing gene. If the gene is upgraded and determined to be a disease-associated gene, then we at the lab side can go back and revet all of the variants that were previously reported out as variants of uncertain significance and turn those into potential genetic answers for the family. And so that is kind of the lab perspective and our lab's workflow. I hope this is helpful information for you. My co-presenters are going to tell you a little bit more about how this looks from the clinical side, but I hope that I have the opportunity to assist all of you or even collaborate with all of you on projects in the future. Thank you.
Yeah, and so as Rebecca's getting her slides up, I want to encourage all of the attendees, if you do have questions, you're welcome to throw those into the chat. Speakers, don't worry about trying to answer those midstream. I'm going to collect those and then we'll have a nice Q&A session at the end.
Can you all see that slide, and is it in presenter mode?
You're good in presenter mode. Thanks.
Okay, great. So as Michelle was saying, there's this sort of side from their laboratory side of this workflow with GeneMatcher, and then it sort of comes to the clinician side. So I'm going to speak a little bit more about that process and just maybe a specific, although very generalized case in referencing this article. I'm sort of talking about that workflow. And talk a little bit more about that. Again, I'm Rebecca Littlejohn, I'm one of the genetic counselors. I work at the Children's Hospital of San Antonio. So how did this article come about? So back in March of 2018, so we do some numbers, that's about 3 years ago, well, almost four now. We received an exome report with a gene of interest or a candidate gene for one of our patients. And we informed the family about the results. So we called them and they came in and we talked to them about these results. And this particular family had been trying to find an answer for their child's health concerns and always expressed a willingness to participate in research. After reading the articles cited in the report, I was personally convinced that this was a very good lead. This gene was very interesting compared to the patient's clinical symptoms and what had been reported about it sort of already, and where the gene and the protein sort of worked in the body. So I reached out to one of the authors listed in one of the publications to get a better understanding if they had any ongoing research at the time, since they had done some in vivo and in vitro models in that publication. We went back and forth for a little bit, but for some reason, one reason or another, that communication didn't continue, and sort of that opportunity didn't work out. Well, 4 months after that communication, I received a contact through GeneMatcher, and another researcher was interested in this gene. So we started to communicate back and forth about this gene, this sort of case series with these patients. We answered some follow-up questions for them. We talked about details of the study, ongoing research questions, and we communicated all of that with the family. We filled out mostly a clinical table with information on a spreadsheet to send to the researcher. Well, about so this communication was ongoing until March of 2020. Was it March? Sorry, April of 2020 when COVID happened. And we were informed that several research labs had shut down because of COVID and a lot of factors that were affecting sort of the research community. And the data was delayed on some of the studies they were doing. So it wasn't until about 3 years after this sort of communication with this group and all of it really came to this publication that you see here. I finally, after reading through some of the communications, actually noticed that that first researcher I had reached out to is actually on this publication. So there was two aspects to sort of tackling, this particular gene, my interest in trying to get the family interested in the gene was that initial contact with one of the researchers through an article that had been published, and then through this contact. So there was sort of two ways that this ended up coming about, I think in a sort of not direct route. So, as you hear this story, you probably have a lot of questions. Why this family? Why this gene? Why did it take so long? And where do you find the time? So why this family? We see a lot of families in our clinics, all of us. And what is it that really, makes us sort of go through the process for specific families? So I've been really fortunate that I've had a lot of continuity of care with the patients in our clinic. And so it's allowed me to build a lot of relationships with our families. To have any hesitancy about research and what it really looks like, I can give them a pretty detailed description of what the process would be and what we're expecting. And I think families really like that clarity when they're considering research. For this particular family, their story stuck with me because they have been very persistent about getting a diagnosis for their child and advocating to say that they want to do research to do that. And they know that that would potentially lead to a diagnosis. And not all families are there to advocate and to actually say that, verbally to their clinician. So I kept this patient on my list of patients because of their particular set of health issues. And that just seemed very genetic, and also the family's interest in participating in research. Why this gene? We get a lot of exome reports and a lot of reports with variants and genes of interest. It was just something when I read those articles and I just started going down the rabbit hole of saying, this is really interesting. I think that there's something there, and I wanted to explore more. And I've contacted a lot of researchers through articles, through gene matchers over the years, and asked if they were doing ongoing research. Some led somewhere and some didn't really lead anywhere. And it can seem intimidating, but I often remind myself is, I have nothing to lose just to ask. It doesn't stop, like asking and getting an answer of no is, we just move on to the next thing for the families. And a lot of the researchers I've reached out to are really eager to help. They really want to grow the research in the area, and they know we're all working towards a common goal of finding an answer for families and just moving forward the science of genes and our understanding of them. 3 years sure is a long time. It's a very, if you think about it, it can be a very long time. I've worked on cases that probably most of them were closer to 2 years. And I've been fortunate enough to work in the same practice for a while. So again, I have that sort of continuity with the research cases and the patients. I work with great colleagues who are willing to work together to collaborate and get these studies done and move forward and find a diagnosis. And then I guess probably the other question I'd propose was, where do you find the time? a lot of clinical work, a lot of other projects or activities you're involved in, whether that's with, you know, the genetic counseling programs or other types of teaching that you do, you know, why this, how do you find the time? We have a very busy clinical practice. That's most of what we see, and my time is dedicated towards. But I think the process makes it very easy, you know, contributing to an Excel spreadsheet with clinical information that you already know about the patient is, you know, I guess a little easier than some other types of research that are out there. You know, a diagnosis is really important to our patients, ultimately leading hopefully to changing their medical management or even individuals in the future. And, you know, when we work in clinic, that's what we're looking for, is how do we affect change in a patient's healthcare? And, you know, without a diagnosis, it's my opinion, without a diagnosis of some kind, particularly genetic, patients can't go towards a treatment odyssey that's very specific to their needs. So, you know, even if we're the only people trying to find an answer for our patients, moving in that direction of trying to find a diagnosis has no deadline to me, and even if it takes 3 years. So I'll stop there and let the next speaker go. And I'll stop sharing.
Okay, hi everybody. Can you see my slides in the right view? Okay, I see some heads on. So hi again, everyone. My name's Elisa Blesson, and I'm going to be sharing a case of gene matcher and the type of research we're doing at Kennedy Krieger in action. So I have some slides just to help me keep on track. So I work at the Kennedy Krieger Institute in Baltimore. I work in clinical neurogenetics, so I do not have any dedicated research time as part of my role. Really, I'm in clinic doing clinical things pretty much every day of the work week. In terms of our team at Kennedy Krieger, so we have our neurogenetics clinic is what we call this. At the time of this case that I'm going to share with you guys today, we had 3.5 genetic counselors, 1 person part-time, and just over the pandemic, we've grown into 6.5 GCs. During this case, and really a lot of the gene matcher collaborations that we've been part of, we had 0 genetic counseling assistants. Now we've grown to 2. And truly our clinic is a little bit unique in the sense that we do have your typical medical clinical geneticists, but we actually mainly work with neurologists who do not have board certification in genetics. So our role as the genetic counselor is really to have the scope of the genetics role in counseling as part of the joint appointment with a lot of our patients. I would say our patient population is highly diverse, whether it comes from socioeconomic status, ethnicity, diversity, education levels, which is one very interesting thing about our population. We do skew towards more pediatric patients, but do have a substantial number of adult patients that come and see us. And the unifying theme here is, even though this is neurogenetics, our scope is really focused on individuals with neurodevelopmental disorders. So common phenotypes or diagnoses that we see are autism, intellectual disability, developmental delay, epilepsy. And Kennedy Krieger, even before I started, was an early adopter of using clinical exome sequencing and ordering it for our patients since 2012. So we've had a lot of experience collectively as a team with getting exome results and oftentimes getting candidate genes, just like Michelle mentioned earlier. So the case that I'm going to walk you guys through timeline-wise, just like Rebecca did, this took many years to happen in the end, is we had a family with 2 affected full siblings. So we have a 3-year-old male and a 2-year-old female. And the interesting thing is both of the siblings presented with this similar history of global developmental delays. So they were late to walk, late to talk, late to hit all of those major developmental milestones. They had low muscle tone or hypotonia, and then the male sibling also presented with dystonia. So we had this interesting phenotype. We had 2 siblings, full siblings in the same family,
So our approach was to start off with exome sequencing. So we used the brother of the family as the proband. We included the sister, and then of course we had mom and dad there. And the exome came back in 2017, and we didn't get a hit for anything at that time. But instead, we had this result of compound heterozygous variants of uncertain significance in a candidate gene, just like Michelle shared earlier, called SPATA5L1. And you can see from the pedigree on the top, both siblings had those same two variants, and each variant was inherited from one parent. So this is shaping up to be an interesting story, but of course, like GeneDx noted on the report, GeneDx is the one who did this particular test. There is nothing in the literature from a clinical standpoint about what this gene meant or what to think about a phenotype associated. It was truly unclear at that time. So the following year, we actually get an email from the lab saying, hey, we have 2 researchers that are studying this gene. Would you like to be introduced to them or potentially collaborate? And we said yes, because the family at the time when we disclosed the exome result had said, you know, if you learn any new information, please let us know. And we typically see our patients and families at least every couple years in case there are updates like this to share. And so through that email communication, we learned that these 2 other clinicians had patients who had the same neurodevelopmental phenotype, so developmental delays, intellectual disability. And so still trying to learn more, and this is still an unclear result. On the family's end, we now learn that the younger sister has an epilepsy diagnosis, and the family tells me, hey, we would really like to think about having more children, and asked, are there any updates about this genetic test result? And again, we said we're collaborating or trying to see if there's anything out there, but at that point in time in 2018, we had no concrete evidence one way or another. 2019 comes around, and this initial email thread that started with 3 of us or 3 clinician groups here has now grown, where we now have 15 more individuals with this similar neurodevelopmental disorder phenotype. So this is getting more and more interesting. And one of the clinician teams as part of this email group says, hey, we're going to take the lead and we're going to organize a case cohort series with a plan for publication. And one other group at our institute is going to take care of some functional studies. So they reached out to all of the groups on the email and said, hey, would your patients be interested in sharing medical records and or potentially sending us skin biopsy samples to help with some of those functional studies? So this family actually had a follow-up appointment with us that coincided nicely. The timing of this helped a lot. And we talked to the family saying, hey, there's this group, they want to study this particular gene more. 1, are you able to sign a medical record release so we can send them medical records to help with the case cohort series description? And then 2, are you interested in submitting skin biopsies? And they actually asked for skin biopsy samples from both kiddos and mom and dad. And this was a really highly motivated family. And I think that's one of the reasons this is a successful case, is they said yes. For a lot of families, the thought of doing a skin biopsy, a skin punch biopsy, may not be the most entertaining idea, and a lot of families end up declining, but this family said, hey, we want to contribute any way we can. And actually the group that led the publication had their own IRB protocol. So I didn't have to consent them. I didn't have to go through the risks of a skin biopsy with them. The group that took the lead with publication did all of that through their own IRB protocol. We just happened to schedule it to happen at our institute where one of our geneticists did the skin punch biopsies. And the family also let us know that because this result was still unclear, even though it was shaping up to be interesting, that they decided to pursue another pregnancy through IVF with donor gametes, just because they were so scared of that potential recurrence risk if this was indeed the genetic diagnosis here. So I think this is where the pandemic comes into play. And like Rebecca said, a lot of these academic centers got hit where their labs and studies had to be delayed, but a lot of the written efforts, so writing up these cases, ramped up. So in 2020, through that same email thread that's been going through since 2018, they created an Excel spreadsheet. So nothing fancy, nothing special, literally an Excel document with just yes or no boxes. Does your patient have global developmental delay? Do they have XYZ symptoms? And so this was easy for us to fill out because we knew the clinical summary of our patients from sending Exome. So we filled out that Excel spreadsheet. And as we submitted and as more cases are shared in this Excel spreadsheet, we realized, hey, our number has grown to 28 families, and hearing loss seems to be a more highly penetrant part of this phenotype, which our patients up until this point had not had there. But really the neurodevelopmental phenotype of developmental delays, hypotonia persisted here. So then the exciting piece happened in 2021, where there is a publication that included our family here of biallelic variants in SPATA5L1 leading to intellectual disability, spastic dystonic CP, epilepsy, and hearing loss. And you can see just from the title how much this overlaps with the phenotype of our patients here. And so it was actually a pretty emotional and somewhat exciting result update visit with this family to tell them, hey, you contributed to the literature. And yes, this seems to be the answer for your family too. And the thing that we did from a medical management standpoint is we said we need to be checking for hearing loss in these kiddos as they continue to age. And in fact, at the point in time of the publication or shortly after, we realized that these two kiddos had some mild hearing loss, and the plan is to continue to monitor them annually with hearing exams to make sure it doesn't get progressively worse or intervene if we should need to. And the parents were so excited about this that they said, hey, we might be thinking about IVF with PGT in case they want to have another child who doesn't have these two compound heterozygous variants. And so we did receive a new clinical report from the lab. So originally it was a variant of uncertain significance in a candidate gene. And now we have a nice, pretty report that says positive. These are pathogenic variants in a disease-associated gene. And that's been very helpful too, not just for our family, but I also got an update just a few months ago that the parents of these patients, they're the oldest siblings. So they have their own brothers and sisters, and now they're sharing their report with their siblings so that they can do their own kind of carrier screening to see if they also have one of these variants for their own family planning purposes too. So I think this is a nice view of how this one collaboration really helped an entire family and their extended relatives too. And that's why I like this particular case, especially when it comes to gene matcher and research collaborations, because it's a nice relationship between clinical practice and research where we ended a diagnostic odyssey for this family and we were able to help with medical management. There's not a cure, there's not a treatment directly for these variants, but we knew, hey, we need to look out for hearing loss or seizures and things like that. And truly for this family, this was their major driver, is thinking about expanding their own family and that recurrence risk counseling, not just for themselves, but for their extended relatives. And one nice thing that often happens out of these collaborations, and I think is still in its infancy in this particular case, is the families create their own Facebook group or family support organization. And that's really an opportunity for them to connect with one another for a rare genetic diagnosis, to know that you're not alone or isolated in that case too. And these cases are one of my most exciting opportunities to not only end a diagnostic odyssey, but also contribute to the medical literature. A lot of us that collaborate through GMatcher will have a co-authorship here, but also just expanding the knowledge about another rare genetic diagnosis, I think is helpful for all players in this realm. And at the time of the publication, this was a group of 28 families. And what I anticipate is as more people do genetic testing, exome sequencing, we're just going to find more individuals with this particular diagnosis. So if there are any future case cohort publications or longitudinal studies, it's nice that this family is plugged in the right way, or we're plugged in should we have another case come up too. A couple areas for consideration for all of us in this meeting here is this was easy enough that it was an Excel spreadsheet and I didn't have to worry about IRB protocols to do skin biopsies or share information. But one thing that, as our team has grown and we now have genetic counseling assistants, is rather than the GC spending the time filling out the clinical tables or clinical summaries, can our GCAs go through that clinical note and help with filling out those types of forms or communicating with the collaborators and family to figure out what the next steps are? I do think having the buy-in from the family and the interest and the motivation is essential for this process to work. Some families are very interested just by nature of genetic testing, knowing that if we do exome, there's no guarantee for a clear result, that they're interested in anything they can contribute to help give some clarity to these types of results. And I think that was essential for this case, especially. And then one thing I always like to keep in mind, especially, is accessibility. So not all of our families are able to do exome sequencing. Not all of our families have access to researchers or clinicians that connect them in this way. So I think this is an area of improvement for all of us in this community too. And that is it for me. I'll turn it back to everybody else.
All right, thank you so much to all of our speakers. We really appreciate you being here. And I'm going to just pull up the questions from the chat. We have some other questions. Julia, did you have any that you wanted to start with?
Well, first, I just want to thank all of the speakers for sharing this experience. And I think I think some of us might have been aware of GeneMatcher and kind of how it comes to be. One of the things I was really kind of struck with by the speakers is just how, you're doing research, but you're doing research to advance your clinical care. And it was really important to be involved in research in this way so as to not only kind of have a direct and immediate impact on the patient care that you're providing, but also kind of help. It certainly is going to help to move forward the research that is needed to kind of in future for your current patient and future patients to have more advanced care. The one I was curious about is that all of these cases kind of extended for a significant period of time. And I was curious if any of the speakers could speak towards kind of how do you navigate kind of keeping track of this case over this extended period of time, and kind of who's the owner? Who do you feel is like the owner of keeping track of that? Is it upon the lab that did the analysis? Is it you? Is it the family? Kind of who kept that going, and how did you organize that?
So in our clinic, it's the genetic counselors. Whenever we get so I would say typically when we send off genetic testing for an exome or any testing for, we have our geneticist, one of our geneticists is on the report, as well as a genetic counselor is on the report. And we divide up those cases amongst the genetic counselors in our team. And they're responsible for following up on it and following up with the families. So, and we keep track of it. Our EMR allows us to have, we call them patient cases, but open cases for an extended period of time, which includes open research cases. So I know other people do it a different way, maybe have internal databases that they use to sort of track families and track ongoing research. That's how we divide it up. How do you?
I would say it's very similar, that it's on the genetic counselor, or geneticist, or physician. We do recommend, especially if it's a non-diagnostic exome result, that the family follows up with us at least every year, every couple years. So that's a nice opportunity to check in to see, hey, last time we talked about this opportunity, here's what we know that's new, or here's what you can do to participate if you're interested. It is hard when the families are lost to follow up. Those are the tricky ones. But I think the nice thing about the pandemic, in a sense, is everyone's become a little bit more flexible with ways of communication, whether it's email or not my chart, but EMR portals where you can direct message families, or phone call, traditional methods, letters, things like that. But it is on the genetic counselor and kind of our genetics team to keep track of where things are. And this is another area where we feel like our GC assistants might help us keep track of collaboration timelines too.
From the lab end, I agree that the when it comes to the care of an individual patient, the kind of responsibility for checking in on the collaboration status is going to fall on the clinical team. We're from the lab end, though, whenever a new variant comes to our attention, or whenever a new inquiry about a particular gene comes to our attention, if we haven't heard anything from the research team, the leads on the research team in a while, we will reach out to them and say, hey, how about an update? And so we try to keep our internal records, if not totally up to date as possible. And we also field questions from counselors who are involved in projects, asking, hey, have you heard anything about this project? I'm going to be seeing this patient shortly, and I'd like to give them an update. And we can take those kinds of questions too and try to get to them in a timely manner.
Okay, perfect. Thank you. So we do have some questions coming in the chat. One of them from Colleen Kellishu is, can the speakers please address the equity issues around family selection? I worry that if we prioritize families who advocate for research participation, we may be leaving families with less privileges and resources behind.
So I can start with this question. So I wouldn't say that we prioritize the families or select them. So our approach has been, if we get an exome result and there's a candidate gene and the lab contacts us, that there is an active collaboration, or through Gene Matcher we get those matches, we make a point to share that with every family. And we tell them, what are the expected next steps, whether it's, do you agree or consent to sharing records? And that's when it's kind of on the family to say, and we talk through this, it's not a yes or no question. We present all of the opportunities that we're aware of depending on the results to the family, and let them decide what they are willing and not willing to do. And there's definitely some concerns about research and how much information to share. And so that's why I think we make a case to have that personalized, tailored discussion anytime we see this type of collaboration opportunity pop up. We don't have a way that we say, oh, if the family says yes to all research opportunities, they jump the line in terms of time or effort too. But this is also an area that, and this is what I meant by that accessibility area of consideration, a piece that we are actively considering and thinking about as we move forward too.
We have the same or similar process where any interest in a gene, we reach out to every single family and we let them know there's this research, potential research collaboration that's going on, and try to follow up with them as much as we can to really talk them through the process if they're interested in it, or if they just outright say no, just say, well, maybe in the future we'll figure out something else. So I don't know that it just so happened that I think it was more so that the gene of interest came up for these patients that we had, and then they were also interested in research. But we do have patients where genes of interest come up and they haven't really expressed as much as these families had in research, and we still approach them as well. And many families are willing to try to participate, even though they hadn't initially expressed interest. They see, they're very altruistic. They see the benefit of having this information and sharing it with other medical professionals and getting this information out there.
I think that it brings up a great point that that not everyone has access to the type of genetic testing that might spur these invitations to collaborate, or might allow a family to have an opportunity to participate in research. Most of these opportunities come through exome testing, and so the more individuals and families who are able to access that kind of testing, the more opportunities they'll have in the future.
Yeah, basically getting any type of test approved, whether it's a genetic test or any type of medical testing, is based on your insurance and or what type of payer system you're going through. And that is complicated and something that probably, for our population, we have a highly, a heavy, a large number of patients who have Medicaid. And it's a complicated system, and probably need some advocating to your legislator about it, or at a different level. So yeah.
Okay, and then another question came through. This is from Karen Wayne. I had a colleague once who shared with me that after their first experience like yours with contributing to case series, they felt forever changed because it felt so good and they were more open to making the additional efforts. Would either of you be willing to share how you personally felt after your experiences, and did this change anything for you professionally?
This is, I think I said this too, one of the most exciting parts of my job as a neurogenetic and pediatric adult genetic counselor is helping families with diagnostic odysseys, and whether it's, through a candidate gene collaboration that ends up becoming disease associated, or if it's, a positive result that comes out through the beginning. Just that ability that we have to help our families through their diagnostic odysseys and finally being able to give them answers, or, having those ongoing conversations it's what helps me get through every single day as a peds genetic counselor, when sometimes you get bogged down by lack of access to genetic testing, or insurance denying the test 3 times already and having to wait for policies to change. I think it's very rewarding being part of these collaborations. And it's something that I was aware of as a student. And because Kennedy Krieger had already been doing this before I started, it was already a part of my role, or my own plan as part of what I do. But it's nice to have that positive reinforcement that what we're doing is really making a difference, even if it is just filling out an Excel spreadsheet sometimes too.
I often tell people that I worked in a laboratory doing basic science research for a very long time, but I feel like in my time at GeneDx, I've actually done more to move research forward than I was able to do in a much longer period of time, toiling in the lab. So it's a really, I feel good about the work that I do.
Yeah, I mean, I think, if we were to put ourselves in those patients' shoes, and we were to say we have our child is having difficulties, and how I think we would want someone to advocate for us, and to say that it's important that my child gets the diagnosis. I want to figure out what's going on, and to just really reflect on that, and try our best to help out as much as we can. I find it really it's just a really fulfilling experience to do something like this, and to change their medical management. I mean, they wouldn't have known that information before. I mean, it wasn't out there. How would they ever find that out about the hearing loss? I mean, it's just, they just wouldn't know it. So it's something small, but it's actually something very important to that family, and changes their child's management significantly.
Okay, thank you. And another question came through. So this is from Jessica Feldman. Excel sheets were mentioned several times in regards to their usefulness for collaboration. In the clinical realm, presumably there is a single general Excel sheet with all patients with candidate genes, as opposed to separate sheets and documents trying to distinguish between phenotype and gene right from the start. What data did you choose to collect in the spreadsheet, and how did you decide to include or not include that data? So for example, did you include one column for major clinical symptoms versus minor symptoms, or individual columns for each body system, or something else entirely?
So I actually think it's even easier in that if this ends up being a case cohort series, usually the group that's leading the efforts creates the clinical spreadsheet themselves. So they create the columns, whether it's by organ system or, major clinic symptoms, other symptoms, and then they email it out to the group. And so my job would be to see what data they're asking for that they've already created there, and then just input from reading the clinic notes or what I know about our patient's phenotype, just filling it out our column of that larger Excel sheet. So I think it's even easier that we typically don't put all of that effort in the beginning. We wait for the collaborator to say, hey, here are the things that we're interested in writing up. Here's what it looks like in an Excel spreadsheet. And here's where we want you to enter your column of information too.
I would add that,
Go ahead. Sorry.
yeah, I was just going to add that if you're asking about how to set up one of these, there's a lot of great examples out there in the literature. If you look up a particular gene, like you've got a couple of examples from this webinar, if you look up these papers, and you'll be able to look at the clinical columns and see what types of things are being asked for. If you were interested in like being the person who pushes one of these projects forward.
And I would say that the columns the collaborators come up with often have to do with like what type of question they're trying to answer. So for my patient, it was like a neurological phenotype. So I asked a lot of questions about their neurological phenotype maybe over other body systems. So that's just something else to keep in mind.
I have a question, and just because we're coming to an end, I want to make sure also before I ask the question that I have time to thank and acknowledge Kristen Hansen, who's leading the webinar group and made great efforts to put this wonderful webinar together. And thank you to all the speakers. It was just so interesting and valuable to hear your experiences. I'm curious to hear from other people who are here too, if anyone, we've heard this great model of how you can be involved in research as a clinical GC using GeneMatcher, and I think that's really inspiring. And I'm curious if anyone else has found other ways to get involved in small or not as small research efforts in using a different way other than GeneMatcher. Like, has anyone kind of had success just cold contacting authors of papers or posting on a SIG and getting a group of people who are interested?
So I can actually, I've received emails from clinicians who have asked about getting in touch with authors of particular publications, or if someone has contacted us asking about, like, is any research going on this gene, we might actually be able to put them in touch directly with the lead authors on those other, on a previous paper, that kind of thing.
That's a great idea, just to contact labs, because they would have a lot of information about who's interested in those genes.
Hi, everybody. Thank you for such a great webinar. My name's Tiffany, and I work at the University of Iowa. And just to answer your question, I've had a lot of success reaching out to authors of papers, particularly if they are doing functional variant analysis already in the gene that you're looking at. And then I've also had a lot of success reaching out to the laboratories and asking them to connect me with other people that have the same variant.
Great. Well, thank you again. And yes, Emily, thank you for recognizing the work that Kristen Hansen has put into this webinar. We really appreciate it. And thank you everybody for the great discussion and hearing about these cases. As always, feel free to reach out to us about this and be on the lookout for future announcements about our next webinar in this series. So thank you again for everybody's time. Have an excellent weekend.